So now we're going to go deeper into the different kinds of helper T cell, like we talked about at the end of the last video. This image just going over on the left, the activation of naive T cells when they come into contact with their antigen, an antigen that their T cell receptor can pick up on an antigen presenting cell. And then when these naive T cells are exposed to these different cytokines like interleukin 12, interleukin 4, interleukin 1 beta, they'll become one of these three different kinds of helper T cell, which all serve different functions, shown in this image. So getting into these functions, we're going to start with Th1 cells, which target intracellular pathogens, like viruses or mycobacteria. These Th1 cells activate phagocytes with interferon gamma, resulting in infected body cells being phagocytized by phagocytes like macrophages or neutrophils. They also stimulate class switching of B cells to produce immunoglobulin G antibodies. We're going to talk more about class switching in a different one. It's also called isotype switching. It's basically just different components of immunoglobulin molecules, which is just the fancy word for antibodies. It's kind of similar to exon switching that we covered in the genetics module. It results in kind of different components being put together to produce different kinds of antibodies which serve different functions. Th1 cells also produce other cytokines which recruit immune cells to the site of infection. So you can imagine interferon gamma is resulting in infected body cells being phagocytized. So you can imagine that these other cytokines are going to be recruiting phagocytes like neutrophils or macrophages. And like we said before, they mainly deal with viral and intracellular bacterial infections. Moving on to Th2 helper T cells. These activate B cells and stimulate them to produce immunoglobulin E via isotype switching. These immunoglobulin E antibodies opsonize helminths, which are little flatworms that we've talked about before. Another interesting thing, these immunoglobulin E antibodies actually bind to mast cells, which we talked about when we talked about the cells of the innate immune system. And remember, when an antigen binds to one of these IgE antibodies on the mast cells, it stimulates degranulation, and these mast cells end up excreting molecules like histamine, which triggers a hypersensitivity reaction. Basically, histamine it'll cause stuff like rhinitis, or like sneezing and runny nose, basic symptoms, to try and get rid of any pathogens on our mucosal membranes. But if we end up producing immunoglobulin E antibodies that have variable regions complementary to harmless antibodies like harmless antigens like pollen, then we're going to end up with a hypersensitivity response and an allergic reaction to pollen, and then you're going to get hay fever. Th2 helper T cells also secrete interleukin 5, which activates degranulation of neutrophils to help kill cells, because remember, neutrophils are granulocytes, as well as phagocytes. They also secrete interleukin 4 and interleukin 13, which activate macrophages to produce enzymes for collagen synthesis and fibrosis. This can lead to granuloma formation or tissue remodeling. So If you remember when we talked a bit about mycobacteria in one of the earlier videos, we talked about formation of we talked about that with respect to mycobacteria producing these granulomas on the lungs in tuberculosis. Now finally, Th17 helper T cells, they defend against extracellular bacteria and fungal infections by secreting interleukin-17 and other cytokines which recruit neutrophils which can phagocytize bacteria and fungi. Th17 cells are also particularly important for intestinal barrier integrity, and they play an important role in the development of autoimmune diseases, which we're going to talk about quite later in this playlist when we go on to autoimmunity and systemic lupus erythematosus. So now these were all different types of CD4 T cells, so different types of helper T cell. Now we're going to talk about the function of the CD8 cytotoxic T cells. So cytotoxic T cells have the function of killing infecting cells. They detect an antigen on the MHC class I receptor of an antigen-presenting cell. That indicates that the cell is infected with an intracellular bacteria or virus, and then the cytotoxic T cell responds by making this antigen-presenting cell undergo apoptosis. And remember, we talked about here, you can see the image on the left, the cytotoxic T cell. The T cell receptor is complementary to the antigen. You can see the CD8 co-receptor, and then the antigen presenting cell is presenting the antigen on MHC class I. The cytotoxic T cells, they kill antigen presenting cells by inducing changes within the cell. They can do this by releasing the contents of granules into the antigen presenting cell cytoplasm, for example perforin or granzymes, which degrade the antigen presenting cell's membrane, and this ends up with the cell dying. Or the interaction of these things called Fas molecules expressed on the antigen presenting cell's membrane. It's basically just receptor binding, giving the antigen presenting cell an apoptotic signal to kill itself. Cytotoxic T cells are specific to a particular pathogen because, as you can see here, the T cell receptor on the cytotoxic T cell can only recognize one kind of pathogen if it's the same shape as this the region of the T cell receptor that is in contact with the antigen. That distinguishes the function of cytotoxic T cells in the adaptive immune system, which produces a specific immune response to specific antigens to create resistance to specific pathogens, to the next cell that we're going to talk about, natural killer cells, which are also descendants of the lymphoid stem cell lineage, but they do not have a role in the adaptive immune system. They have a role in the innate immune system because their response is non-specific. We're going to talk about why that is now. So natural killer cells are activated by a target cell not presenting MHC class I on its cell surface. And then if it doesn't find MHC class I on a target cell surface, then it'll initiate it to commit apoptosis. They can also do a thing called antibody-dependent cell-mediated cytotoxicity, where natural killer cells will bind to antibodies bound to pathogens via the CD16 Fc receptors Upon binding, the natural killer cell will release granzyme B and perforin to degrade the membrane of the pathogen and killing it. So it's a non-specific response it's killing. It doesn't care about an antigen, it kills everything.